here's our ribosome. So if this whole thing is a cell, this whole whiteboard is the cell, this is all cytoplasm. Here's a ribosome down here. This guy has to get to that guy. So it has to leave the nucleus. So the final step here's the nucleus. Here's your DNA that's now seals itself back up. Here's your RNA strand. So what are we going to do to it to terminate it, to process it, to get it ready to move? What did we say we were going to do with it? There's 3 things we do to finish it up. You want to remember? What do we put at the front of it?
The
5 cap. That's because this is the 5 end, so we put a 5 cap on the front. What do we put on the back?
The poly tail.
The poly A tail. And what do we do at the middle? Again, remember, your code includes a lot of stuff. And when that RNA polymerase was reading the whole code, it's reading some important stuff and it reads some not important stuff. Important stuff, not important stuff. It's kind of like you guys doing homework. We'll kind of skim through. That looks important. Let me read that. Wait, that's not important. I won't read that. So there's little pieces where it doesn't read the whole thing, the whole message. So what you're going to do, as you remember, is there's going to be pieces that are important. We call those exons. And there's pieces that are not important, called introns. Okay, so which one do we keep?
Exons.
We're going to keep the exons. X for exit. So exits out, introns stay in. So you will do what's called splice. Enzymes in your body will splice those guys out. And then all of the exons, all the important stuff, squeeze chemically, just bond back together. And you end up with a finished RNA strand that contains only exons, important stuff. And then the pre-mRNA now becomes the mRNA, and you're done. The first step is finished. Okay? How was that? Make sense? We're okay? Yeah. The mRNA is going to leave the nucleus now. So after it gets so this was the termination and processing of it. It got the 5 cap, the tail, and it's only exons, important genetic material. And now it's going to leave the nucleus. Okay, so that's part 1, transcription. All right, so translation then picks up right where that leaves off. Here's the nucleus. Here comes your mRNA. It leaves the nucleus. Where does it go? Ribosome. R for ribosome. It will come around, and ribosome has 2 places, the upper and lower part, so it's going to go right in the middle of it there. So this guy will actually come right through here. And this is now reading it, much like that RNA polymerase read it. This is now called the rRNA is what makes a ribosome. So now this will read this code again, and we're going to flip it one more time. So as this guy is reading it, it calls upon, I should have made this a little bigger. Let me make it bigger. Okay, so there's the mRNA, and it's got codes in 3s. So we'll just make this easy. CCC GGG. It's got 3 codes are called a
Codon.
codon. So every 3 base pair are called codons. This ribosome reads, as it's coming through, it's going to read them in groups of 3. So the first one that came through is this guy right here. It reads CCC. And coming through this thing all the time are tRNAs. So you have all these guys out here. These are tRNAs. And each of them have an anticodon, a 3 base pair. And they're all different. So they will keep coming through until the perfect one that has GGG, this one maybe has CCC. This will come through, doesn't work, goes out. When one comes in that has the perfect code of GGG, it'll form a bond, because there's your purine and pyridamines, there's your G's to C's, perfect match. Locks on, and then as it continues to move and it leaves the ribosome, this guy will leave. And it's got this amino acid on the top right here. This is our amino acid. So as this guy continues to move and it leaves, that goes away, and all you're left with is the amino acid. Okay, so let me stop there. Everyone look at that and make sure that made sense to you. Okay, so here comes our mRNA, codon, 3 base pair. tRNAs are coming in. When one fits perfectly, it latches on. That means that knows that it's needed. There's the amino acid that we've coded for. We need that one. So as it continues to move, this guy goes away. We don't need it anymore. And you're basically left with just a bunch of amino acids. Those will form a bond between them. And Michelle, you were gone for this, but remember how amino acids bond at the amino group on one end and the carboxyl group on the other? So they form bonds. The bonds they form are called peptide bonds. That's a peptide bond. And then this is basically going to turn into a protein. Okay, so if you get enough of the peptide bonds, they call it a polypeptide chain or a protein. So this is our protein and we're finished, it's done. And that part was called translation. So the first step was transcription. That was transcription occurs in the nucleus. It basically took a DNA and made an mRNA.